Welcome one and all to another episode of Southern Crop Sciences Seminar. Bringing you two of the department's finest speakers to lead off the student presentations. And Dr. Stelly, would you like to make an introduction?
I would. So a few new faces out there. I'm Dave Stelly in the Department of Soil and Crop Sciences, and it's my pleasure to introduce one of my students, Christian Hitzelberger. Christian came to us from Dallas, and he basically approached us after he completed his bachelor's degree up at Sam Houston in plant soil science. One of the things that attracted me to him as a student was he had conducted undergraduate research on Bermuda grass clover, is that right? With Bobby Lane. So he had already had, evidence of interest in plants and whatnot. So yeah, it's been a pleasure to have him as part of our program. So he joined us and worked with us for a little over 2 years on his master's degree. Joined us originally in 2017, and during that time he worked on some of what he was going to talk about today, because he carried through on the research with chromosome segment substitution lines, which is a form of interspecific germplasm integration. And when he finished up his master's, we talked about it, and we rolled him into a Ph.D. program where Dr. Jane Dever, who's up in Lubbock and is a bona fide applied plant breeder with very different kinds of expertise than I have, so highly complementary. So we're co-advising him for his PhD program. Of course, he's going to talk about that today. And so I'll let him do that. I will mention it's always kind of fun to know a little bit something personal about people. So I asked him to give me a couple tidbits about himself. And so one of the things that's kind of interesting is he's had for, 8 or so years, he's had a silversmith, an engraving type of business, which he continues to run. And then as additional hobbies, he likes to fly, and he likes to fly fish. So he does both of those things. So interesting. But anyway, Christian, yeah, let him have it.
Cool. All right, thanks for the intro. All right, so let me start this. Keep track of that. All right, so Dr. Stelly mentioned me talking about part of my research today, which is the fiber characterization of cotton introgression lines. Here's a quick overview. So some of the current issues were limited genetic diversity in upland cotton, all while trying to maintain and improve economic yields for the global market, because cotton is an export crop. So we're exporting pretty much everything we grow in the States. And then we're also trying to increase crop improvement progress. So to address these issues, our lab specifically is doing that through the development and characterization of introgression panels. Specifically, those are the chromosome substitution lines, which I'll be talking about today. And we also have chromosome segment substitution lines. And some background on cotton, for those who aren't familiar with it. Cultivated cotton is a tetraploid, so we're working with 52 chromosomes. The main two species that are cultivated are Gossypium hirsutum, also known as upland. Makes up about 96% of what's produced in the United States, and this is your basic kind of blue jean cotton. The remaining is Gossypium barbadense, probably heard it as Pima or Egyptian cotton, makes up about 4% or the remainder that isn't upland. So like I just mentioned, we've got limited genetic diversity in upland. This kind of started a long time ago, 1 to 2 million years ago, with a monophyletic tetraploid origin. We had two diploid genomes come together to create a tetraploid. And we have multiple studies showing low allelic diversity in hirsutum. And then that bottlenecks even further in the 1980s with the trend of reselection for proprietary materials. And more recently, we're studying large haplotypic blocks that exist within the genome with preferential transmission. So as an example of that, this photo on the right is chromosome 8. It's kind of blocked here, but just for the gist, chromosome 8, the red sections are haplotypic blocks. So those areas of the chromosome are inherited as one piece. So as breeders, you can obviously tell that can be a problem if you're trying to separate genes. Variation is available, but progress is slow. So if we can improve that, obviously, that's preferred. To give you an idea of kind of the phylogenetic tree here, we have diploids, and then 1 to 2 million years ago, like I said, some A&D genome-like ancestors came together, which gave rise to the tetraploids. In the tetraploids, the two cultivated are Hirsutum and Barbadense, and then the species we're specifically using for these chromosomes, Barbadense, Tomatosa, and Muslinum, introgressed into Hirsutum. So now getting into the introgression panels, it's really just incorporation of wild germplasm or just other cultivated germplasm into our cultivated cotton. And so why create these introgression line panels? Why not just evaluate the wild germplasm as it is, right? We get this question a lot because these panels are a backcross derived population. So this comes up a lot. The main reason is that these traits that we're after are really complex. There's genome-wide interactions, right? There's a lot of genes involved, and there's epistasis as well. So we really have to dissect that wild genome to evaluate its potential. It's hard to detect anything positive when you have the noise of a lot of negative alleles in the background. So this photo is just an example of the cotton seeds in a bowl. On top is Hirsutum, and on the bottom is a wild species. So you can see that that bottom wild species is not agronomically viable as it is, and it'd be pretty hard to evaluate the fiber quality of that right there. So chromosome substitution lines, it's kind of a scary name, but it's pretty simple. It's just a collective representation of the wild genome across a panel of lines, which is a number of lines here. So the differences between the lines are whole chromosome or chromosomal arms. So you look at the figure on the bottom here, you have kind of 5 example lines where each line has a different chromosome, the red representing an introgression and the blues that upland background. And that background is near isogenic. I'll kind of talk about the isogenic platform next, but it just means that it's all Hirsutum background. The only thing that changes is the introgression. Everything is the same Hirsutum background. And these are created through multiple generations of backcrossing, but it requires an aneuploid Hirsutum parent to create the chromosome substitution line. That's how we introgress a whole complete chromosome. So that's kind of a limiting factor in creating a entire set that has every chromosome, is because we have to have that aneuploid parent. So a little bit about the isogenic platform. Really, this is, we have these multiple species I've talked about introgressed into the same Hirsutum background. So when we compare everything, it's really an apples to apples. The only thing that's changing between the lines is that introgression. Versus if we had introgressed into multiple Hirsutum backgrounds, where you're going to get a lot of different interactions. But with these, we can intermate and do a lot of different crosses, and everything stays the same except the introgression. So it really allows for that apples to apples comparison there. So I want to talk a little bit about cotton fiber, what it is and how it's measured for the people that aren't familiar with it. So cotton fiber, it's mostly cellulose, single elongated epidermal cell from the seed. Each seed can have up to 20,000+ fibers on a seed. So as it grows, it's just basically kind of think of it as a tube or a straw. And as it matures, cellulose is deposited inside that initial cell in different orientations. And when that fiber matures and dries out, the way that cellulose is laid down in different orientations kind of pulls on the fiber, and you have a twist, right? And so these twists actually help in creating yarn, because that creates a little bit of friction between each individual fiber and helps the yarn stay together. So this is just some fiber I picked out of the field, put it under the microscope, took a picture. And this is just a thread I pulled out of my shirt, so you can kind of get an idea of the finished product, how all these fibers are now aligned and kind of twisted together. And that's kind of the end product there. And how are we? The industry standard is HVI, high volume instrument. So every bale that's produced in the US is measured on this system. You get micronair, which is essentially a relationship between how thick that fiber is. Yeah, we'll just say that, not to get too in the weeds with it. And then length, you get a measurement of the upper half mean length of your sample. And you get a strength, which is a bundle tenacity. Really, you're not testing individual fibers. This machine takes a number of fibers at once, clamps them, measures the length and everything, and then pulls it apart. And where that breaks is your strength. How far it stretches before it breaks is your elongation. And uniformity is just related to length, how uniform your fiber length is. And we also have Aphis, Advanced Fiber Information System. It's a little more advanced, a little more expensive, not as fast, takes a lot longer per sample. But it's an individualized fiber tester. So you're testing 15 to 20,000 fibers within a sample. So you get a lot more resolution, which was important for this project because at a chromosome level or chromosome arm, we were kind of expecting to see small, minute differences. So being able to really dissect that fiber quality was what we wanted, and that's why we ran both of these systems. But you get a length distribution by number, by weight. You can see the top 5% of fiber length, short fiber content, which is a really important indicator for yarn quality. That's just the percentage of fibers that are half an inch or shorter. More short fiber quality you have, the lower quality your yarn will be. So that's an important one. Then we get maturity and fineness. I kind of talked about how cotton starts growing as a tube. You can kind of think of maturity of how thick that cell wall is, how thick that tube wall is, and then fineness is kind of how big that overall size is. You get a lot of others, but like I said, I'm not going to talk about them all today to spare y'all and myself. But into the experiment, we've got 2 years in College Station. We ended up with 59 total chromosome substitution lines with 1 recurrent parent in there as well. So going to RCBD, 2 blocks. We had 5 plants at each line. And then all that was, of course, surrounded by guard plants to eliminate any spatial advantage. And then the whole plants were harvested. All that fiber was bulked and stored it for about 2 weeks to dry and let it equalize the humidity, because different humidity levels in the fiber can kind of give an advantage or disadvantage. So I just wanted it to be all similar, so there was no advantage at the gin. And these were all ginned on this tabletop saw gin you see here. And then the fiber was sent to the Fiber and Biopolymer Research Institute in Lubbock at Tech for HVI and Aphis analysis. And this is really just here. This is a lot, but just gives you an idea of, these are the different lines we had per introgression. So it kind of gives you a relative idea of what lines we had representing each species. We have the most for Barbadense because Barbadense is well known for having high fiber quality. So that's the one that's been worked on the most. And our recurrent background is an upland hirsutum, Texas marker 1. It's pretty standard, genetic standard. So everything's been introgressed into TM1. And as I move through these results, I just want to kind of explain this label. You'll see this label a couple of times, but it's pretty simple. It's just a letter followed by a number. The letter represents the species, and the number is the chromosome number that's introgressed. So MO7, this is, okay, I don't know. Where's this? There. So M7, chromosome 7 from Muscellinium introgressed into hirsutum. A B22LO, which means the chromosome 22 long arm of Barbadense introgressed, and T16 short, chromosome 16 short arm of Tomatosum introgressed. So you'll see a kind of combination of long arm, short arm, or whole chromosomes from different species. But just want to give you that, kind of understand what's going on. So into the results. done these by species, so it's a little easier to digest instead of doing everything at once. So our barber density that we've introgressed is Pima 379. It's a genetic standard line. So with Aphis average length, we had 11 significantly different, but they were all shorter than TM1, which was 0.79 inches. And if you're familiar with cotton fiber length, you're probably wondering what's going on with that. That's really short. We have to remember this is Aphis, and Aphis takes into account short fibers where HVI doesn't really. So you kind of have a more fair average in Aphis, but it's just one thing that Aphis gives us. And if we look at Aphis and HVI next to each other, so we're looking at average Aphis length on the right, the top 5% of fibers in the middle, and HVI upper half mean length on the left. You can see that upper half mean length and the top 5% of fibers in Aphis produce the same results there as far as significance, both showing that barber density 25 and barber density 14 long were significantly different from TM1. Barber density 25 has been previously reported as an improved fiber line, so it's nice to verify that. And then moving on to another important quality was that short fiber content I mentioned earlier, which is the fibers that are half an inch or shorter. So in barber density, we had 7 significantly different from TM1, but they are all higher. We didn't have any that were better with barber density. So TM1 was 20.5%. And those that were significantly different ranged from 25.4 to 31.5% short fiber. So one of the questions we have to ask here is this due to mechanical breakage where the fibers are being broken and we're seeing an inflated short fiber content value because of, handling it too much, or it's being broken in the gin, or as it's harvested, or is it a genetic effect? So one way we can look at that is by I want to start off here with actually the fiber distribution. So on the x-axis here is fiber length bins. So there's, I think, 20 or so bins here of a certain fiber length, and the y is just the relative frequency. So you get a distribution here of the fiber lengths. And remember, this is 15 to 20,000 individual fibers that are tested, measured to get this distribution. And so our short fiber content, barber density 3 and 4 are some of the high. So you see those graphed there in kind of the salmon and green, and then 8 long was not significantly different, but it had a pretty unique distribution profile. So I included on there as well. TM1 is in the black, that's a recurrent parent check. So how we ask that question, is it breakage or is it genetic? We can look at maturity ratio and fineness. For maturity ratio, you're looking for something that falls in 0.8 to 1. That would be your average maturity. Less than 0.8 is a little immature, and over 1, you start to get into very mature. And so that's just how thick that cell wall is. And then fineness is, again, how thick, how fine or coarse the fiber is. So if you're going to see breakage, you're either really high maturity ratio and really coarse, or really low maturity ratio and fine. So kind of our results here were, it seems we may be getting a little breakage, but it might be mostly genetic. It's also important to note cotton fiber doesn't have a linear density. So this is kind of the average fineness and maturity. So you could get kind of tips of long fibers that aren't as mature or are too fine and are breaking off and giving you that average change in fineness. But moving on to our next species, Musselinum. So wild species endemic of northeastern Brazil. Got a really pretty flower there. And so with Aphis, kind of the same story, the average length from Aphis, we had 5 significantly different. They're all shorter than our recurrent parent. When we look at upper half mean length and the top 5% of fiber lengths, we actually found kind of differing opinions between HVI and Aphis top 5%. So in the Aphis top 5%, it said Musselinum 1 and 4 were significantly longer than TM1. But in HVI, muscellinium 1 did not pop up while 4 did. So it's kind of interesting to see some short or some long stuff come out of this wild species. And then with short fiber content, we had 9 significantly different and 3 were actually significantly better. So this is pretty exciting because showing a second, but so we had muscellinium 17, 22 long, 26 short, all having less short fiber content than TM1. If we look at this graph, you're going to start to see a trend, obviously, with short fiber content. You're going to have that tighter distribution, ideally above ½ inch. But muscellinium 4 with 28.3, you can see how different this kind of flat distribution is compared to the other short fiber content lines and TM1 in there. And if we look at maturity ratio and fineness, M4 is a little fine, so we might be getting a little breakage there. But with the other 3 low short fiber content lines, they have pretty average maturity ratio, and their fineness is within we're looking for kind of 170 to 200 as an average fineness. So they all look pretty good. And what's interesting here is actually M17 in green is almost identical to TM1 as far as maturity ratio and fineness. So these would make really good improved fiber quality lines if we could kind of move that average length more. But it's interesting to find this in our introgression lines. Brings back the point, dissection of that genome is necessary. So this is the photo from the beginning showing that a recurrent parent here and that introgression parent. So you really have to dissect that genome into pieces to effectively search it and find things like this short fiber content trait. And so our last species was Gossypium tomatosum, wild species endemic of Hawaii. And with Aphis and HVI length, all the lines were significantly shorter, or they weren't significantly different from TM1 in both Aphis and HVI. We didn't really have anything beneficial come up as far as length with tomatosum. But with short fiber content, we had 3 that were significantly different, and one of those was significantly better. Again, so we had tomatosum 16 had 14.3% short fiber compared to TM1. Again, this is a really important yarn quality measurement. And again, just kind of showing these graphs to give an idea of what these distribution looks like. Chromosome 2 had 29.1%, while tomatosum 16 had 14.3%. And what's important here is the maturity ratio and fineness. Maturity ratios look pretty standard, and the TO2 is a little fine, but it shouldn't be so fine. It's not too fine that we'd expect a lot of breakage there. So this is most likely some genetic effect influencing this. Obviously, T16 maturity ratio is okay, fineness is all right. And so through these results, we kind of had some introgressions that kind of were common among our linked and short fiber content. So within our length, we had a number of what I'm calling common introgressions. So we had chromosome 10, 5 long, 8 long, 11 long, and 16. So what's important here is it's most likely not the presence of the introgression that's affecting the fiber quality, but more likely what's not there anymore. TM1 came from a really old delta pine line, so obviously it was bred, and whatever was there is probably doing a good job at doing its job. And so we just took out what was probably working. And so that's most likely what's going on there. But with short fiber content, it gets interesting here. We had 4 and 16 pop up, but with 16, barbadensis 16 had a negative effect, meaning it increased short fiber, while tomatosum 16 decreased short fiber. So if we compare those on our fiber length distribution again, we got B16 in red, tomatosum 16 in blue. We just looked at tomatosum 16, so it looked familiar. We're looking at the maturity ratio and fineness to see if barbadensis 16 is having any breakage issues. It's not, it shouldn't, because it's nearly identical to TM1, which is fairly interesting. But T16, again, we looked at that just a second ago. It's definitely outperforming it as far as short fiber content wise. So wrap it up. In conclusion, it's really preliminary study showing that we have found differences that exist across these materials, these chromosome substitution lines. And I kind of mainly focus on the positives to keep it short, but we have a lot of negative effects that are kind of popped up as well that are just as useful as the positive effects. Obviously, the positive effects are program or anything else. But the negative effects kind of give us insight as to what's happening with that trait when we see it dip down so much or decrease or any changes like that. So really the next step with this material is to, we know usefulness exists here and we need more resolution. So what that means is kind of breaking up those chromosomes into smaller portions of the chromosome. So we have chromosome segment substitution lines that are being developed. So that will be kind of a next step, or we can take this to top crosses or CS RILs to kind of narrow down what is actually influencing that trait on that chromosome. So, okay, just want to thank my committee, Dr. Dever, Dr. Stelly, Dr. Rooney, Dr. Sean, Dr. Udall, all the members in our lab that have helped with this, past members who've helped, everyone at the AgriLife Research Center at Lubbock who's helped a lot with the fiber quality, folks at the USDA here in town who help us plant every year. We wouldn't have plants if it, we wouldn't have plants in the ground if it weren't for them. Also Cotton Inc. for their continued financial support through this. So I can take questions.
Questions?
How did I calculate the maturity ratio in the chat?
We'll get to that.
Thank you for your presentation. Super cool. Just where's the relationship between the changes in quality and yield itself? I mean, you see any relationship on that? And it's positive, negative, it's something that
Between the what?
the yield on the field.
Oh, the yield?
Yeah.
We haven't done any, so this has just been fiber quality. We do have a study this summer coming up where we'll do more yield with a subset of these to look into yield. But usually it either goes into fiber quality. It's kind of a, you know, you have limited resources with the plant, right? So you kind of got to balance it out.
Other questions?
So how much does the environment then affect fiber quality?
The environment affects fiber quality, depends on the trait, the quality. Usually length is pretty heritable, pretty set, but something like maturity is pretty dependent on environment, and maturity is kind of a really important part because it can affect a lot of different fiber quality traits. Because maturity is set over multiple weeks, it can take a lot of hit from the environment or positive effects from the environment. So it's just kind of depends on the trait.
Yeah, it's in the chat.
Yeah. How do I calculate maturity ratio? Aphis, the machine, does it for you. It's got electro-optical sensors that measure that, and that's an output from Aphis.
By science.
Yeah, science.
Other questions?
Can I speak? Okay, great. Interesting talk, thank you. So you were mentioning the giant haplotypic blocks in the beginning, and then you have a chromosomal substitution line. If you were going to make the RIL from those, do you think that the recombinations, or lack thereof, is going to be a problem there?
Yeah,
What's the plan?
So that's, I didn't mention because it's not my project, but one of the things our lab's working on is working to break those haplotypic blocks up. And so, yeah, I don't know. I think that's all I can say without speculating wildly.
Don't ever do that. You get yourself in trouble. Let's thank our speaker again.